Hi, my name is Carlos Portera Cayo, and I'm a professor of neurology and neurobiology at UCLA. And I'm happy to be here to tell you more about this project that we just published in the journal Neuron. Just to briefly introduce what we do in my lab is interested in understanding the causes of symptoms in neurodevelopmental disorders. And in particular, we focus on a disease called Fragile X syndrome. And in this particular study, we identified a protein that is important for Fragile X syndrome, and we think it might be a potential therapeutic target to consider in the future. Is EPAC2 different, and are we more excited about it? Yes, because EPAC2 is a target that we found in an unbiased screen for gene dysregulation in Fragile X mice. And we were looking particularly at cortical neurons. And the gene expression studies pointed to EPAC2 as being the only gene that was dysregulated both in excitatory and inhibitory neurons. It was increased, the levels were increased in those cell types. And also, it's the only gene that is highly enriched in the brain. So other genes and proteins might be expressed all over the body, but EPAC2 is very enriched in the brain. It's also a target of FMRP, the protein that is missing in Fragile X, and FMRP regulates a lot of different proteins and genes, so EPAC2 is one of them. And the last reason is that mutations in the gene for EPAC2 have been associated with neurodevelopmental disorders in the past, including autism. And the other reason why we're excited about EPAC2 is that when we reduce the levels of EPAC2, we found that it rescued not just circuit differences in the brains of Fragile X mice, but also multiple behavioral deficits in the mice. Things ranging from susceptibility to seizures to memory problems and also what we call tactile defensiveness, which in children with Fragile X relates to an avoidance to what is considered an aversive tactile stimulus. There's sort of a sensory hypersensitivity phenotype. So all these are all the reasons why we thought EPAC2 would be exciting. The protein that is missing in Fragile X is called FMRP. It stands for Fragile X messenger ribonucleoprotein. And this protein is expressed all over the body and presumably in all the cells in the brain. And one of the long-standing theories of autism and other neurodevelopmental conditions is that there is an imbalance between excitation and inhibition in the brain, and an imbalance that favors too much excitation. And the thought was when you're missing a protein and you're a neuron, and you're either an excitatory neuron or an inhibitory neuron, when you're missing this protein FMRP, does it predominantly affect the excitatory neurons or the inhibitory neurons, or does it affect them equally? One might think, well, FMRP is important, is a synaptic protein that, or it's a protein that is expressed highly in synapses, and it might do the same, it might play the same roles in excitatory and inhibitory neurons because they all have dendrites and axons. They both have neurotransmitters that they release and synaptic vesicles. So you might think that FMRP might play similar roles. We actually found that when you're missing FMRP in these Fragile X knockout mice, the gene dysregulation was very different in excitatory versus inhibitory neurons. So that was a bit surprising. And it could explain why there's actually this imbalance between excitation and inhibition. Now, for the purposes of EPAC2, that gene was dysregulated in a similar way in both cell types. And that's why we thought it would potentially be a good therapeutic target. But the surprising thing about the paper, I suppose, is that when you consider excitatory and inhibitory neurons, which are neurons and they just, one releases glutamate and the other cell type releases GABA, but generally they're both neurons. Why would FMRP play such different roles in each cell type? And our data suggests that, yeah, it does play very, very different roles. Yeah, it's hard to tell because, first of all these discoveries were made in a mouse model. And symptoms of even social behavior in mice are quite different than the way that humans interact, and that the types of maybe social anxiety that boys with Fragile X might manifest. So we think that particularly the sensory hypersensitivity phenotype rescue that we discovered with this EPAC2 antagonist may be something to focus on potentially for translation in humans, because a majority of boys with Fragile X experience sensory hypersensitivity, particularly to tactile stimuli. And so the fact that this drug rescued a lot of the aversion to repeated whisker stimulation might potentially translate to improving sensory hypersensitivity in children with Fragile X. Yes, thank you for asking that question. Indeed, EPAC2 we found was upregulated or overexpressed in adult Fragile X mice. But in fact, in other previous studies that we had done, we checked for EPAC2 expression in younger mice at age postnatal day 15, which is equivalent maybe to the infancy or toddler stage. And at that point, we did not find EPAC2 to be upregulated in Fragile X mice. Now, this means that, clearly EPAC2 expression changes even across development in neurotypical brains, um, but that in Fragile X, it may only be upregulated in adults. This is actually good news for Fragile X syndrome because there's a concern that during development, the trajectory of typical maturation of the brain somehow deviates in autism, including in Fragile X syndrome. And some concern has been that maybe if you try to intervene in the adult or in older children, maybe it's too late because now the brain is in a different sort of state or regime, and it's hard to reverse whatever happened during development. But the fact that EPAC2 is upregulated in adult Fragile X mice and that we intervene by inhibiting it and could rescue a lot of phenotypes in adult mice suggests that potentially it could be a mechanism for intervention in older children and adults who live with Fragile X, and that it is never too late to intervene in this disease. The main message is of renewed hope. As we said earlier, there's been a lot of failed clinical trials that did not reach endpoints, and this has been very disappointing, including some really recent results of a big clinical trial in Fragile X. And even though this comes with a lot of disappointment, there's also a lot of lessons that have been learned from these failures that will help in clinical trial design in the future, patient selection for clinical trials. And EPAC2 is just a new target, and it's just one example of dozens of other targets that are being discovered every year by basic scientists across the country and around the world. So my message to pediatricians is to communicate to patients that there's a lot of really smart people working in the labs around the world to discover new treatments for Fragile X, and that EPAC2 is just one example of a potentially a target for the future.